Hi, I'm Taupp from the laboratory of Stephen Hallam in the Department of Microbiology and Immunology at the University of British Columbia. Today we'll show you a procedure for the construction of a large insert environmental library. Assisting me in today's procedure is Sangwon, who will be performing phage packaging and infection of the library host E. coli, as well as Alyse Hawley and Jinshu, who will be performing the colony picking. We used this procedure in our laboratory to study microbial community structure and metabolism in marine and terrestrial environments. So let's get started.
This protocol for creating a fosmid DNA library begins following isolation and shearing of genomic DNA from biomass samples, procedures that we have detailed in accompanying JoVE articles. The next few steps prepare the sheared DNA for ligation into the fosmid vector, PCC1. The first step is to repair the ends of the sheared pieces of DNA and phosphorylate the 5' ends. All reagents necessary for this step are included in the PCC1 Fosmid Library Production Kit from Epicenter. To set up the end repair reaction, thaw all reagents on ice. Then combine the end repair buffer, DNTP mix, ATP, up to 20 micrograms of sheared insert DNA at 0.5 micrograms per microliter, enzyme mix, and water to reach 80 microliters. Briefly centrifuge the tube to get all liquid to the bottom. Incubate the end repair reaction at room temperature for 45 minutes to an hour. Then heat and activate the enzymes at 70 degrees Celsius for 10 minutes.